So today's our topic is about how the acidic drugs absorbed in the human body. When we are saying that acidic drug, then it means that those drugs whose pH are less than the 7, means their pH might be 5, might be 6, might be 4, might be 3, might be 2, might be 1, means their pH will be less than the 7. If their pH is close to 1 or 2, then we can say that this is very strong, this is very strongly acidic drug. If the pH is 5 or 6, then we can say that this is weakly acidic drug, means it is acidic in nature but it is not very acidic in the nature, it is weakly acidic. So if the pH is between 4 and 5, then we can say that it is between the strongly acidic and weakly basic acidic pH. So in this scenario we have to study the example of the weakly acidic drug, and that weakly acidic drug is aspirin. So aspirin, what is aspirin? Aspirin is a weakly acidic drug. Aspirin is a weakly acidic drug. When we are saying weakly acidic drug, then we can also explain how it is a weakly acidic drug, which characteristic make the aspirin weakly acidic drug. We know that the pKa of the aspirin is approximately 3.5, and we also know that the what is the pKa. Ka, in this part, the Ka is the dissociation constant of the acid. What it explains means, greater the dissociation constant of an acid, greater will be its acidity, means it is directly relation to the acidity. Greater the dissociation constant of the acid, greater will be its acidity. But pKa, what is the pKa? pKa is the negative logarithm to the Ka, means negative logarithm to the acid dissociation constant. So what is pKa? pK is negative logarithm of the dissociation constant, and it relates with the acid or acidity as inversely, means it is inversely proportional to the acidity. So when we are saying that there is an acid whose pK is very large, then this means that if its pKa value is very large, then it will be less acidic in nature. But when we are saying that its pKa is less, then we can conclude that its acidity will be large, means it is more acidic in nature, means its pH will be closer to 2, 3, 1. If we are saying that its Ka value is high, then it means that it will be strongly acidic, means if the Ka value is high, the acid will be strongly acidic. So this is overall about why aspirin is a weakly acidic drug, because its pKa is 3.5, and the 3.5 pKa is not very much small, so it makes it acidic, but this acidic is not very much. This is weakly acidic. This is our main concern, that aspirin is a weakly acidic drug. Now if a person swallow aspirin drug, what happens? How this aspirin drug absorb in its body, and how this aspirin drug reaches to the target tissue where it functions. So the main whole mechanism in this scenario is, when we swallow an aspirin, the aspirin will go into the stomach. From the stomach it passes the membrane, it crosses the membrane. How it crosses the membrane, we will study it. And it, after crossing the membrane, it enters into the blood vessels. From the blood vessels, through the blood, which is the transporting material, transport it to the specific tissue from where it will perform its specific function. So how this mechanism occur, how the aspirin from the stomach is how the aspirin is absorbed or enters from the stomach to the blood vessel, how this process occur. So this is our main concern, this is our concern area. So what happened, consider this is the stomach, and a person swallow this drug, and this is the aspirin. I am saying this is the aspirin. If I am saying this is the aspirin, then we should come to know that aspirin is a weakly acidic drug. And why it is a weakly acidic drug? Because its pKa is equal to 3.5. that's why aspirin is a weakly acidic drug. And we know that the pH of the solution, in this case, in this scenario, what is the solution? In this scenario, this stomach, the stomach we draw here is the solution. So we are discussing about the pH of the solution. We always discuss about the pH of the solution, and pKa of the acid, and pKb of the base. So pH of the solution, pKa of the acid, and pKb will always for the bases. So this is the aspirin, and we ingest this aspirin, a aspirin, and whose this is a aspirin, and whose pKa value is 3.5. And we know that the pH of this solution, in this case the solution is the stomach, the pH of this solution is equal to the, means the pH of this solution stomach is equal to approximately 1.5 1.0 to 1.5. This is the approximate pH of the stomach. Now what you can conclude from these pKa and pH value? What you can conclude? You can conclude that the pH, which is 1.0 to 1.5, is less than the pKa, which is 3.5. Or we can conversely say it that the pKa of an acid, which is 3.5, is greater than the pH of the solution, means which pH of the stomach, which is 1.0 to 1.5. So what happens if the pKa is greater than the pH? So you can see that if the pH is less than the pKa, or pK is greater than the pH. In this case you can see that the pK is greater than the pH, or pH is less than the pH is less than the pK. What will happen? Acids and bases will exist in their protonated form. Means if the pH of the solution is less than the pKa, or pKa of an acid, or pKb of a base, then the acids and bases will exist in their protonated form. In this case, we already know that the protonated form of an acid will be uncharged, whereas deprotonated form of an acid will be charged. Why we are saying this? Because suppose this is the acid, suppose this is the aspirin, which is a weakly acidic, this dissociates and form the hydrogen ion, and this A negative its conjugate base. So you can see that this is the protonated form of an acid, because a proton is attached to the acid, while this is the deprotonated form of an acid, because the proton has been washed away from this conjugate base. So what this scenario suggests, in which the pH is less than the pKa, or pKa is greater than the pH, this suggests that the acids and bases will exist in their protonated form. We are talking about acids, so our concern is about this side. So acid will exist in their protonated form, and we already discussed that the protonated form of an acid will be uncharged. So in this case what will happen? This uncharged form, means what will happen, this uncharged form of an acid, which carry no charge, while this charged form of an acid will be present abundantly in the stomach. What it means? This means that in this whole scenario, this protonated form or uncharged of an acid will be abundantly present than the deprotonated or the charged form of an acid. Means if the aspirin is 100% of the aspirin is present, then 80% of the aspirin will exist in their protonated or uncharged form, while 20% of the rest of the aspirin will exist in their deprotonated or the charged form. We can't say that the 100% of the aspirin will exist in the uncharged form or the protonated form. We can't say this. Approximately maximum of the aspirin will exist in their uncharged form. So what it suggests, what is the benefit of the aspirin being abundantly uncharged or protonated in the stomach? What is its advantage? So its main advantage is that this is the stomach, and in the peripheral of the stomach there will be some blood vessels. Suppose I am drawing this as the blood vessel. This is the blood vessel in which the blood will flow. This is the blood flowing in the blood vessel, and we know that the major component, one of the major component of the blood is the plasma. One of the major component of the blood is plasma. This is the blood. One of the major component is plasma, and we also know that the plasma contain approximately 92% of water. A plasma contain approximately 92% of the water. What it suggests? Water is a polar molecule. Water is a polar molecule which makes the plasma polar, and the plasma makes the blood polar. So what will happen? Blood contain plasma, plasma contain water, water is polar, water make plasma polar, plasma make blood polar. So we can say that the blood is a polar medium. Blood is a polar medium. So we can say that this blood is a polar medium. So we said this, that the blood is the polar medium. Now what will happen next? How this this acidic drug aspirin has to pass into the blood, why this aspirin should have to pass into the blood? Because through the blood, which is a transporting medium, it will be transported to its specified site. What is the specified site? The site at which it will perform its function. Means the site at which is specified for to perform the aspirin function is the specific site or the target site. So blood is required for it. So that's why this aspirin has to diffuse into the blood. And how this will diffuse into the blood? After crossing the stomach wall and after crossing this blood vessel membrane, this blood vessel. And we know that these stomach wall as well as the blood vessel, all the cells, stomach cells, blood cells, all the cells contain a membrane around it, and that membrane we call as plasma membrane or cell membrane. And we know that the plasma membrane do not allow charged substances or large substances to pass through it, while small substances like and non-polar substances like the carbon dioxide or lipid soluble substances can cross the plasma membrane easily. Lipid soluble substances, small substances and non-polar substances can cross the plasma membrane easily, because plasma membrane is also non-polar, so non-polar can cross the non-polar medium easily. What is the advantage of this abundance of the aspirin in its protonated or the uncharged form? The benefit is that this protonated or uncharged form can cross the cell membrane easily. Means this cross this cell membrane and this cell membrane easily, and can get into the and can gain entry into the blood vessel easily. So we studied that what is the purpose, why the nature made this this pH of the stomach's acidic. So the purpose of this is that the weakly acidic drug can cross the membrane easily and get entry into the blood. So when they get entry into the blood, what will happen next? I am drawing this blood vessel here. This is the blood vessel in which the blood is flowing, this is the blood flowing, which contain plasma as main constituent, plasma which make 55% of the blood, and other white blood cells, red blood cells, platelets, etc. And we conclude that the blood is a polar medium. So in this case of the blood, what if we are talking about the pH of the solution, then it is clear that we are talking about the pH of the blood. So in this case, the pH of the blood is, we know that the pH of the blood is equal to the 7.4. This is approximate pH of the blood. So what will happen after this acid in the form of the aspirin or the weakly acidic drug, more in the form of the protonated or the uncharged form, passes the cell membrane and get entry into the blood vessel? What will happen? This is the this is the aspirin which get entry into the blood vessel, and here this is the aspirin same, and we show this aspirin as HA. So this HA will ionize to form hydrogen ion plus its conjugate base A negative. So what this whole scenario suggests us? We know that we already discussed that the pKa of the aspirin is approximately 3.5, which shows that it is weakly acidic. So in this case, you can see that the pH of the solution, in this case the solution is the blood vessel or the blood, pH of the solution is 7.4, whereas the pKa is 3.4. This scenario suggests that the pH is greater than the pKa or pKa is less than the pH. Means this mechanism will apply here. Means pH 7.4 is greater than the pKa. In case of an acid, it will be pKa, while in case of the base, it will be the pK. pH will we are talking about the acid because aspirin. pH 7.4 is greater than the pKa 3.5. So what will happen in case of the acids? If in the case of the acids, pH is greater than the pKa, then the acid will exist abundantly in the deprotonated form. And we all know that this is the deprotonated form of an acid, and the deprotonated form of an acid is charged. You can see that this is the deprotonated form of an acid which carries a negative charge. This is the protonated form of an acid which carries no charge. So this suggests that in the blood, the pH of the blood is 7.4, whereas the pKa of the aspirin is 3.5. In this case, the pH is greater than the pKa. This suggests that the aspirin will exist more abundantly in its charged form. In its charged form means, what is its charged form? Its charged form is the deprotonated form. So aspirin will exist abundantly in its this charge form. Means if 100% of the aspirin enter into the blood cell, then 80% of the aspirin will exist in its deprotonated or the charged form, while rest of the 20% will exist in their uncharged or the protonated form. What is the benefit of aspirin or weakly acidic drug to be exist in the charged form or the deprotonated form in the blood? The main advantage of this is that the blood, we already discussed that the blood is a polar medium. So according to the like dissolve like, polar substances will attract the polar substances, or polar substances will be stabilized in the polar substances. So that's why aspirin has to be polar to stabilize, to stay in the aspirin has to be polar to stabilize or to stay in the polar blood medium. So we all discussed about this, how the acidic drug absorbed in the body from the stomach to the intestine. This is the whole mechanism.